Hi there. Today we're going to look at Chapter 11, labeled immunoassays. Let's get started. All right, chapter overview. You can follow along with this as we're going through the chapter. I do have a nice chapter roadmap during the middle of the lecture just to keep us on track. So, if we think back about a couple of weeks ago when we learned about agglutination precipitation reactions, what were we talking about? We were really talking about antibody and antigen reactions that we could detect in patient serum, and then we visualized it in the lab through those types of reactions. We could actually see the outcome of those reactions. Labeled immunoassays are, really very similar in concept in that they're all based off antigen antibody. Unlike precipitation and agglutination reactions, which are quite visible in the form, we saw them precipitation, we saw red cell agglutination, we saw turbidimetric reactions. Labeled immunoassays are things that are being, antigens and antibodies that are either very small in size, so you're thinking something like a hapten, or present in very low concentrations, where you might be thinking of a very early immune response, for example. So they're far too small or too fewer reactions to be visualized in that way. So by labeling some of the test reactants with something that's going to have a reaction, we're not going to be visualizing the actual antigen antibody reaction, but we're going to be visualizing a product of the labeling of the reactant. And we think of this frequently as an enzyme reaction, for example, that causes a color change in the presence of after an antigen antibody has been bound. So it's kind of an indirect monitor that the reaction has taken place, but it's still based on, the base model of antigens and antibodies. Similarly, like flow cytometry, we found that we can label all sorts of reactants with some kind of a marker. And in labeled immunoassays, we're going to look at things like fluorescence and enzymatic markers. Radioactive, chemiluminescent are the ones we're really going to focus on. And once we see the process of these tests, whatever the label is really kind of incidental to that. So we're going to look at the basics of the test methods and then interchange out the labels for different purposes. They all have different reasons to use them. In general, labeled immunoassays really are particularly helpful because we're not going to just determine the presence of an antigen or an antibody, but we're going to be able to quantitate it. And we're going to do it by reading kind of the end products of those reactions, of those labeled reactions, and read it off a standard curve. That you remember how we did that when we created a 3-point standard curve using calibrators. We're going to do the same thing with this. We're not going to go over standard curves again. If you are in any question about that, go back and look at that process that we reviewed when we did precipitation and agglutination reactions. Labeled immunoassays are really mostly widely used to monitor things, bacterial antigens. We're going to see repeatedly hormones, therapeutic drug monitoring. Tumor markers are a fantastic, this is a fantastic methodology for monitoring efficacy of treatment for tumor by monitoring, the presence of tumor markers. We can even look at some specific immunoglobulins when we're doing labeled immunoassays. So let's just take a look and see where we're headed with this. There are some key terms and methodologies that we need to talk about. I'm going to give you a really quick overview now, just so you've heard a couple of terms, and then we're going to go through them one by one. Once you understand the basic technique, we'll be able to substitute in and out. We'll be able to change in antibodies for antigens, fluorescence for enzyme. There's a lot of things we can do to manipulate the test methods. So we'll talk about the specifics in just a minute, but in general, we term the immunoassay reactions as either competitive or non-competitive. That's kind of our most important cut. Competitive refers to adding whatever you're looking for. So let's say, for example, you're looking for an antigen to see if the patient has an antigen. You're going to add both the patient antigen and a labeled known antigen of what we think we're looking for. So if you were looking for HIV, you would probably not use this test method, but let's pretend you were. If you were looking for HIV, you would add a labeled HIV, and then you'd use reagent antibody to see and monitor how much labeled antigen is bound to the antibody versus how much of the patient antigen is bound. They compete. So basically, in competitive assays, a reagent, a known labeled reagent, and a patient unknown reactant are both added to the reaction, and they compete for the target antibody. In non-competitive immunoassays, this really refers to adding an unknown patient sample to a known reagent target. So in this case, there's no competition. So I'm not adding, if I'm adding antigen, patient antigen, looking for patient antigen, I'm not adding a labeled antigen. What I'm doing is I'm going to label the antibody, and I'm going to look for that exact antigen-antibody reaction. So there's no competition, and the concentration is really 1 for 1. So if you bind one one patient antigen to one patient antibody, that's a 1 for 1 reaction, and they're directly proportional. The concentration is directly proportional to that 1-to-1 reaction. So that's competitive and non-competitive. And competitive is just that, competing antigens or competing antibodies. Patient with a reagent. Non-competitive means it's just a sheer antibody antigen, one of which is labeled. After we discuss competitive and non-competitive, we have to really look at kind of some reactions within those assays. And we're going to term the reactions of competitive or non-competitive immunoassays is having either the test method is either heterogeneous or homogeneous in nature. Heterogeneous assays, and we're going to look primarily at heterogeneous enzyme assays, but heterogeneous assays can also be called separation assays, and these are washing steps. So a heterogeneous assay, we're going to see that anytime we have a possibility of forming an antigen-antibody complex, after that reaction has taken place, we're going to wash the sample. You're going to spend a lot of time when you're in blood bank doing washing, so you'll understand this concept more as you do it, but for our purpose, we're just going to remember that heterogeneous enzyme assays all have at least one wash step. Homogeneous assays don't require a wash step. And as a reminder, wash step is the same as what? A separation step. We're going to talk about each of one of these specific techniques in excruciating detail in just a few minutes. So we know the different kinds of lab assays we can do, competitive, non-competitive. Then what are we going to use for labels? So we talked about how there's got to be one labeled reactant within this. So it's either going to be an enzyme, it's going to be a fluorescence, maybe something radioactive, or even a chemical reaction, a chemiluminescent. Most of what we talk about is going to be enzymatic. And so, either the antibody or the antigen in the reaction, one of those two reactants is going to be labeled with something. When we think of The enzymatic reactions, though, I just want to remind us, kind of, from chem days, that we don't. There's actually one extra small step where we have to go in after the enzymatic reactant binds to its object. So if it was an antibody that's bound with an with an enzyme, and it binds to an antigen, and we've got that complex form, that alone doesn't do anything. That just sits there. We have to add a substrate that is going to cause something measurable to happen. So if we remember that a substrate really just is the substance that the enzyme is going to act on. So that's all a substrate really is. And it's that action of the enzyme on the substrate that causes something to happen that we can measure. Without the presence of a substrate, nothing's happening. So if I happen to forget to tell you that there's a substrate addition, there is. I'll try and remember to always say it. And then one other thing we're going to talk about before we moved into looking at our immunoassays, starting with competitive first, let's talk about that wash step and why it's so important. As I said, separation steps or wash step mean the same thing. We see them in heterogeneous test methods. That's by definition, a heterogeneous test method includes a minimum of one wash step, maybe multiple. And the wash steps are really important to remove any unbound excessive components that might interfere with the assay. So let me give you an example, and I think this will help. So in our test sample, we have an antibody that I have labeled with an enzyme, and that's what that little asterisk is referring to, is it means that there's an enzyme bound to that antibody. And so I have patient antigen, and I have reagent antibody that's got that little label attached to it, that enzyme. And I put them into a test method, a test well together. I let them sit for a little while, and I allow complexes to form. Under normal circumstances, anytime I told you when we are forming those antigen-antibody complexes, what do we have to do? We have to rinse afterwards. But why are we rinsing? So if I have these complexes, and I've got that little enzyme there, right? This complex, when I go back later and I add that substrate, the enzyme acts on this substrate and causes what's called a signal change. A signal change is just referring really to that action. So if we have a colorimetric enzyme, a color change is the signal. The change of color is the signal change. So if we have a fluorescent if we have some kind of a fluorescence enzyme, a fluorescence, the production of fluorescence is the signal. And so we'd have a change in fluorescence or change in color. That's all that's referring to. So when we have that enzyme-bound antibody complexed to an antigen in the presence of a substrate, we get a signal change. But oh look, we forgot to do our wash step, and I've got all this unbound antibody, not bound to any patient antigen. We already bound up all that patient antigen here. There's nothing else to bind it to. But I forgot to wash. I throw the substance into the test well, and guess what happens? You got it. There can be signal change. So you end up with not just measuring the complexed enzyme, which is what we're interested in. We end up with all, the complexed plus the uncomplexed, and get you get a falsely increased level of signal change. So it's really important that we wash and we get rid of all of this unbound antibody that's got an enzyme attached. Does that make sense? I hope so. So here's where we're going. We're going to look first, as I said, at competitive versus non-competitive. Remember, competitive is just two things looking, either two antigens trying to compete for the same limited number of antibody sites. Non-competitive, antibody plus antigen makes a reaction. There can be other steps to it, but that's the basics. Non-competitive. So we're going to first look at these two things in great detail, and then we're going to start to look at the immunoassays in a little more specific way as a lab test. We're going to look at those based with radioactivity. We're going to look at enzyme immunoassays and a couple of those very specific ones. And this will be quite, we will have covered most of this up here. So this will be just a quick review. We're going to look at homogeneous enzyme immunoassays. Not very common, but they do exist. Fluorescent and chemiluminescent, and I think in here I also forgot polarizing, which is a really small piece of this, so I might not have really forgotten it. I think I just didn't want to put it. So first up though, competitive and then non-competitive immunoassays. So if we look first at the principles of competitive immunoassays, we'll look at some general information, and we'll talk about it a little, and then we'll look at some illustrations and examples, and we'll see what we can do. Competitive assays almost always usually look for antigen, but the test can be modified to look for antibody. But for the sake of argument, we're going to suggest that the bulk of the time we're going to be looking for patient antigen. As I said, because it's competitive and we're looking for antigen, what else are we going to have within the test method? We're going to have a labeled reagent that's the antigen. So if I'm looking for an antigen here, I'm going to have the same antigen that I want to find, but I'm going to have it labeled. And I'm going to have it labeled, and we're going to use the example of an enzyme. So I'm going to most of the time be discussing enzyme labeling for now. So we're going to have a labeled reagent antigen that's going to compete with the patient sample for the presence of that same antigen, but not labeled, right? It's just a normal patient, literally out of the patient and into the sample. And then we're going to we're going to mix all those things together with some immobilized antibody within the test well and see what happens. Let's see. Labeled antigen is always in excess. This should say in excess right here. Sorry, I will try here. I'm going to put it here just so you have the same thing when you get your, I'll put this back up. Always in excess. Let's see, why do we do that? Because we want all these immobilized antibody binding sites on the test well to be filled up. We want a true competition. We don't want to leave some empty binding sites. So labeled antigen is always in excess, so we know that this test can run to completion. Let's see what else. As I said, the labeled antigen competes with the unlabeled patient antigen for those antibody binding sites. And we're going to see that in an illustration in just a minute. Just remember this for now. It's really important. Competitive assays, always, the amount of bound reagent labeled antigen that binds to that, and we're going to see it in a second, binds to the immobilized antibody is always inversely proportional to the concentration of concentration of the patient concentration. That's wrong. Concentration of the patient. Sorry to be doing this at the same time, but I might as well get it right. Antigen. There you go. Now we have the same thing. Is inversely proportional to the concentration of the patient antigen. What does that mean to us? That means that when I have in that competition, if there was more reagent antigen than patient, I end up with a high level of patient antigen. Control. I end up with a high level of reagent antigen. That means I'm going to end up with, for example, a big color change because there's a lot of enzyme there that's attached to that label. And that means, so a high color change means a low amount of patient sample. We're going to see that a low color change is indicating there's lots of patient antigen in that sample. So they're inversely proportional. Let's look at that in an example in just a second. And competitive, what do we know? It's heterogeneous, so it requires what? It requires a wash, right? Let's look at this test system from page 157 of your book. A couple of notes before we get started. Number one, remember, competitive immunoassays are always heterogeneous, so it requires a wash. Anytime you get antigen-antibody binding, you're going to wash afterwards, right? You're going to wash away the excess. Number two, this particular example is that of an enzymatic reaction. And because it's reflected as a color change here, you can see there's a dark blue, here's a little bit lighter blue. Because it's a color change, we're going to be referring to this really in general terms as colorimetric, but really the enzyme reaction could be a number of different things, or it could be even a fluorescent reaction, it could be any number of things. This happens to just be an enzymatic colorimetric reaction. So let's remember that, but the concepts of the process of competition are going to be the same no matter what. Let's see. The third thing is that we're going to, at the end, measure this against a standard curve. And in this case, the standard curve is showing us the signal is on the y-axis, which is, remember, that's just like a color change, for example. And it's the analyte concentration. And this, because the curve is typically we see it go this way when it's directly proportional. This is indirectly proportional. So let's look at this standard curve really quickly. If I have a high degree of color change and I read it down against my concentration of analyte, what's my analyte concentration look like? Very low. High signal means low analyte concentration. If I go to a low signal, here's a low, let's pretend our signal is this much. We go over here, read it off the standard curve. I have a low signal, but do I have a high or a low analyte concentration? I have a very high concentration of analyte. Inversely proportional. High color change means low concentration. Low color change means a high concentration. That's what I mean when I say that the signal strength is inversely proportional to the amount of the patient analyte in the concentration. We're going to see why just now. Let's see, I think that was all my 1, 2, 3. Yep. Okay, great. Let's look at the test system that we're looking at here and get our feet wet with this before we go too far. So the test system includes enzymatically labeled antigen, and that's reflected here as just these little yellow star things. We have a limited amount. Remember, we're going to have a limited amount of antibody that's immobilized. We call this a capture antibody. Why is it doing it? Because it's going to capture these antigens, right? So this is called the capture antibody. Capture antibody is immobilized to the solid surface of our test wells. It's usually some kind of a test well that we're doing this in, but we're going to immobilize the antibody. Why do we immobilize it? Number one, because we don't want to wash it away, right? But number two, we also want to make sure that we have it so that it's sitting up in the right direction. It wouldn't do us any good to try and capture these antibodies if our these antigens, if our antibodies were upside down, for example, and the FC region was up in the fluid. So immobilizing antibodies helps us to orient the antibodies the proper way, but it also makes it so that once this antigen binds to it, it doesn't get washed away in solution when we do that wash step. We have two different samples here. We have a sample A and a sample B. Sample A reflects a negative control, or we could call it a negative antigen, no antigen, or this would be exactly what it would look like if a patient didn't have the antibody as well. So it's just a negative sample, no antigen of whatever we're looking for in question. Sample B, and the book always uses it uses red as the patient, it denotes red as the patient. So sample B shows us there's the enzyme antigen, the enzyme-linked antigen, and there's the patient antigen. And you can see they're in the sample at the same time. They're going to be mixed together with that antibody, and we're going to let it rip and see what happens. So both of these test methods, so we first start with, let's start here. let's see, what else do I need to just tell you about this before? So I told you about the capture antigen, the capture antibody, negative control. Okay, so let's look at sample A. What do we know about this? So we added patient sample, we added reagent antigen, and we already had the immobilized antibody in the test well. So I added the two kinds of antigen to see if they compete, right? There's no little red dots. So is there any competition for these unbound FAB reagents? There really isn't. Just these antigens are all going to bind off. And you can see, because this is always in excess, we bound off 100% of the antibody binding sites. We just made a complex here, didn't we? We made the antigen that we added plus the antibody. And now they're complex together. We have an antigen-antibody complex. Once that reaction happens, what do we have to do? We always have to wash. I'm going to wash now. I wish the book had shown some free antigen left here so that it would show that you have to wash it off. They didn't. So we're going to wash that. And then what do we have to do? Remember, this is just going to sit like there forever unless I do something. And that is, I need to add a substrate now to this. I have my enzyme here. I have to add the substrate to the enzyme here. I do that. The substrate reacts with the enzyme, causes cleavage, and it causes the formation of color. This one happens to be a colorimetric one. There's an awful lot of antigen here bound to this antibody, so we've got a pretty deep color change here. Let's look at sample B, where there is, in fact, some patient antigen, as well as the same reagent antibody that we added before. Same immobilized antibody. I've added patient sample. I've added reagent antigen to the immobilized antibody. I let it sit. We're going to wait. It's going to incubate for a period of time that the manufacturer has told us how long to let it sit. We do that. We're going to have some competition for these open binding spaces. Some of them are going to get filled, kind of like musical chairs, right? Some of them are going to get filled by patient. Some of them are going to get filled by reagent. If this one happens to be an equal concentration, which again, not the best illustration, but it is what we've got. If one of these is in a higher concentration than other, more of the, they take up more of the musical chair spots while we're incubating until all of the spots are filled. So in this case, they're in pretty much equal quantity. So it's neither 100% filled with reagent antigen, nor is it 100% filled with patient antigen. It's something in the middle. In this case, it's 50-50. So what do we do after we have these complexes formed? Right here, we have a wash. We're going to wash, and then we're going to do what? We're going to add the substrate, because we've got to have a reaction for these enzymes here. So we're going to add a substrate to this. And if you look, this is half as much enzyme here. There's only 3 enzymes here, and there's 6 here. So this 3 enzymes can only act on half as much substrate as the 6 here, right? So we get a lesser concentration of a color change. This isn't the best color change difference. I think the book might be a little bit better, but you get the idea. This is clearly lighter here than here. We measure that color change, and we're going to plot it on the graph. So again, sample A had a very high degree of color change, a lot of signal, and it had a very low, in fact, 0 concentration. The patient had no antigen that was in question. In this case, it was about 50-50, so the signal was about half. You read the signal across, where it intersects the standard curve, and then we draw it down to the analyte, and we could quantitate the analyte if we knew what the key was, what the units of measure were. We don't, because it's a sample, but we can tell exactly what the analyte concentration of these antigens, these patient antigens are. This is a competition method. And so we're going to be competing for the open spots between them. And kind of the concentration of the patient or control, the relative patient or control antigen concentrations are what will help to how much gets bound, how much of each gets bound. So it's definitely a relative step. And remember, the signal change, the signal strength we just saw, it's inversely proportional. High signal change, low concentration, that's inverse. They're one high, one low, right? Low signal change means a high analyte concentration, inversely proportional. I hope that concept of inversely proportional makes sense. If it doesn't, please come see me or send me an email. It's really important that you understand that. So, question 1 for our in-class participation. The immobilized antibody in this test procedure is also termed what? We called that the capture antibody, right? That's going to capture the antigen in the test reaction. So we just looked at competitive assays. A non-competitive assay should start to be easier to review now that we've seen what the concepts are, right? We've seen some illustrations. A non-competitive immunoassay can test for either antigen or antibody, and it's just going to depend on what we absorb onto that solid phase first. Whatever it is, whatever we absorb on, we're going to look for the opposite of it. So if we absorb antigen onto the test well, we're going to look for antibody, right? So non-competitive can test for either. We're first going to, as I said, passively absorb the antigen or antibody, and then we're going to allow an unknown patient antigen or antibody to react and be captured by the antibody. Then we wash. We're going to wash because we're going to get rid of all those unbound antigens or antibodies, whatever it is. And then we're actually going to add a second antibody, this time with a label. So this is going to be an anti-human immunoglobulin that's got a label in it, an enzymatic label for our purposes. We're going to wait, and this second antibody, this anti-human globulin, is going to, the FAB region of this second antibody is going to bind to the FC region of the antibody that was bound. So it's antibody to an antibody, which is unique. The FC region is an epitope. It's an antigenic site for that second antibody. So we're going to add a second antibody that has the label. We've created a new complex. We've created an antigen-antibody-antibody complex, for example. So what do we need to do when we get a complex? We're going to do what? We're always going to wash, and then we're going to add a substrate. Signal change is due to the action of the enzyme labels on the substrate, gets measured, gets read on a standard curve, and in this case, the amount of the label is measured directly and is proportional to the amount of the patient antigen. So if we have a high level of patient antigen, we're going to have a high amount of color change. If we have a low amount of patient antigen, we're going to have a low color change because the antigen is directly binding or even indirectly binding to antibody. So we're looking really for that one-on-one reaction. We're not looking for competition. We're looking for true antigen-antibody binding. We can use all kinds of labels in non-competitive, radioactive. We are going to talk mostly about enzymatic, fluorescent, chemiluminescent, chemical luminescence have all been used as labels. So let's take a look at some examples. This illustration shows three, pretty basic variations of non-competitive immunoassays. You're going to find a lot of variations on this theme, and really, in fact, the book shows different variants of this. It all depends on how the test method was designed. I'm using this right now, not because it's um, following right along with the book, but I really liked the illustration of these reactions. I thought they were a nice illustration for us to follow along with. So we'll see these, and we're going to see how they relate to the pictures in the book, the illustrations in the book, and then we'll go to the illustrations in the book as well. I just like this a little bit better. So let's look at this quickly and then move on to the textbook. The first picture, the first one on the left, is what's called a direct assay. This isn't illustrated in this chapter at all in your book because it's really typically used more for non-immunologic testing, things like histochemical staining. So while it's not really readily used for EIA, enzyme immunoassays, it really is the simplest version of a non-competitive assay that there is, so I think it's a fantastic example. So, direct assay, not typically used for immunoassays, but fantastic illustration. We call it direct because the labeled reactant is directly involved in that initial antigen-antibody binding. So when we say that the reactant, the labeled reactant, is involved in the initial antibody-antigen reaction, we call that direct, and this is a direct assay. In this case, the antigen is present. This is the patient antigen, and it's been bound. So let's say it's a cell, just like you do when you do a gram stain. You heat fix cells to or bacteria, you can heat fix a number of things to a slide, and then you add something to it. So that's what we did. We took tissue, we took cells, we took something from the patient, and we fixed it to a slide. Then what do? We flood the slide with labeled antibody that we expect to be against the antigen that we have in question. This has an enzyme. Once we complex, what do? Always we're going to wash it off. And then we're going to do what? We're going to add a substrate. And we're going to look for a colorimetric reaction. We're going to look for a dye. We're going to look for some change that we can measure. We could look at this under a microscope and see the staining, the histochemical staining change. Regardless, we're going to look for the action of this enzyme on the substrate in this. This is direct because the enzyme tagged antibody is directly involved in the antigen-antibody reaction. This is directly proportional. The more signal produced, the more antigen there was. Straightforward. Because remember, if we had any unbound antibody over here that didn't complex, what did we do? We had washed it away. So very straightforward. The second one is called the indirect assay. This is equivalent really to the antibody capture from figure 11-2 on page 158 of your book. This is slightly different, a slightly different form, because remember I told you we can immobilize either antigen or antibody to the surface, to the solid surface. In this case, this example, they bound antigen. They mobilized some reagent antigen. It can be antibody, and in the book, is it antigen? Can't remember right now. Let's see. Yep, it is antigen. Okay, but it can be antibody here as well. So in this case, they have immobilized antigen to the solid surface, and then we add the patient sample. That's the primary antibody that's from the patient. We look and see, we wait the requisite amount of time for the binding to take place, and after we did that, we formed a complex, or we potentially formed a complex. So what did we do before we add anything else? Exactly, we washed. Then we made a second addition. We added a secondary antibody, another anti-human antiglobulin that's labeled enzymatically. So now, oh goodness, where'd that go? So now we have a complex of an antigen, a patient antibody, and an enzyme tagged antibody. Once we do that, what do? We have a new complex, so what are we going to do? We're going to wash again, and then we add the substrate, and we look for the reaction. the level of the signal is directly proportional to the amount of antibody the patient has. But was this secondary antibody involved, the labeled antibody involved in the initial antibody-antigen reaction? It was not. So it's an indirect assay. Okay? This didn't really link directly to an antigen of the patient. It linked to an antibody of the patient. So in this case, if this was a hard antigen for us to find in the patient, we may look for the product of the immune response. We might look and see if the patient has an antibody to that rather than looking for the actual pathogen. So that's an indirect assay. The third reaction is illustrated, is also, we're going to see this later on. This is called a capture assay or a sandwich. Uses the same techniques as sample B in your book, but using an immobilized antibody instead. So we have a capture antibody, and so remember, this is going to be, it's called a capture antibody because that's what's immobilized to the membrane. We have the antigen that is kind of that crazy antigen. We don't really know what it is. And then we have the patient antibody here. We can test for the patient antibody here, and then we have an anti-human antiglobulin. You also have versions of this where you can have just the capture antibody, the patient antigen, and it looks more like this, that you don't have this secondary thing here. But this sandwich is, we're looking at this, where this is going to be the patient antibody and this is going to be an antigen. And we go from there. So that's a sandwich. We're going to talk more about sandwiches in a few minutes. So we'll take a look at that shortly. This really isn't equivalent to sample B there. We're going to talk about when we talk about sandwich assays. So let's look at the one in the book. This is from your book. This again is on page 158. So these are other examples of non-competitive assays. Same concept, slightly different appearance or procedure. Just like we saw when we did the competitive, sample A is our negative control. Again, it could just as well be a negative patient. Sample B is a patient who has the antigen in question, and we just don't know how much. And this is going to be what we call antibody capture. And in this case, believe it or not, it's because the antigen is going to capture the antibody. So before it gets this term, we're going to continue to look at that, and we'll redefine these just slightly for you as you go along. This isn't a capture antibody. This is an antibody capture. Remember, capture antibody are these that are bound. An antibody capture procedure is when antigen is bound and it captures antibodies in solution. I can't believe they named all of this the same with just different combinations of words, but they did. The nomenclature, that's the way it is, so we'll have to live with it. So just like we saw here, sample A, no antigen. Spoiler alert, sample B has antigen, and we'll look at antibody capture in a few minutes. In both cases, you can see we have immobilized antibody here on a solid surface. We add the sample to the test. Remember, this is non-competitive, so I'm only adding the patient sample, or the control sample. We add a sample. If the antigen is present, it binds to the antibody. When we let it sit for the amount of time, the manufacturer tells us how long to wait. They'll say, let it incubate for 10 minutes, let it incubate for 2 hours. It will tell you how long to incubate. You let it incubate and either form or not form complexes, immune complexes. But regardless, after that time is up, what do? We're going to wash. We're going to wash away any excess. So there's excess antigen here, we're going to wash it away. So we're going to wash it away, and we just end up with the complex. We're going to end up with the immobilized antibody with either antigen captured to it or not. Then we add that secondary, a second antibody, and this is going to bind off to a separate epitope of the antigen. So it's not going to bind to this is not anti-human antiglobulin. This is an antibody that's going to also bind to the antigen, just a separate epitope on it. So we add the new reagent antibody that has a label attached to it. You can see it's got that little yellow label. And this had no antigen here, right? So it's not looking for this antibody, so it's just going to stay in solution. Nothing's going to happen. Remember, there's no antigen here. This, we bound off antigen. This antibody binds to the antigen, and we now have a complex of immobilized antibody, antigen, and then enzyme tagged antibody. We created a new complex after that had incubated. What do? We wash. We're going to wash between this step. We wash. What do we need to do after we wash that time? We need to add the substrate to that enzyme, right? This enzyme here and here. We created a color. This has no antigen antibody. This has no antigen nor the bound or tagged antibody. You have no complex here, so guess what? That solution, the color change never happens. There was nothing there to bind that antibody to this solid membrane. This one did. This did have antigen present, so it caused a reaction. So this we're going to say, remember, this in non-competitive immunoassays, is it directly or indirectly proportional? The color change is directly proportional. So you can see the line is going in the opposite direction. And if we did an example, here's the signal. So if we had a signal of a color change here, let's say it's 50%, and this is about half of, it's about 50% bound, I think, maybe less. I can't really see from here. Let's pretend it's 40% bound. You go up 40% on the signal, and you go across till you hit the line, and then you read down, and you can estimate what your analyte concentration is. In this sample, we had a color change of 0, so at 0, we read to 0, and the analyte concentration is 0. It's directly proportional. No signal change means no analyte. If we went up to a higher signal change than even level B and read it over, and then down, our concentration of analyte is higher. High signal change means a high level of the analyte in solution. Those are directly proportional. Very straightforward. Does that make sense? Great. Hopefully it does. Again, come see me if you don't. The antibody capture that we see, the third column here, is really an illustration, the same as we just saw in the indirect assay. Immobilized antigen is used in order to look for antibody in the patient serum. If it's present, the antigen captures the antibody, binds to it here. So we had immobilized antigen, add patient sample that maybe contains the antibody we look for. If it does, it creates a complex. So we have a step that could potentially complex. So what do? We rinse, we wash, then we add the labeled anti-human antiglobulin. So remember, this is not matching to the antigen, but this is going to tag to the FC region of this patient's antibody. And if that happens, we just created a complex of antigen, patient antibody, tagged antibody. After that happens, it incubates. We wait. We do what? We rinse, we wash, rinse, wash, and then we add the substrate. When we add the substrate, color change happens. We read it off a graph. Bing, bang, boom. You're done. Same direct proportional as what we just saw. So that's non-competitive. Competitive and non-competitive. I think we're in great shape with those two. So let's look at these concepts of competitive and non-competitive assays in each of the 4 immunoassay classes that we're really interested in. 5 classes of immunoassays. We're going to start with the heterogeneous enzyme immunoassay. Oh no, we're going to start with radioimmunoassay. So let's go to radioimmunoassay. When we first look at this, RIA, radioimmunoassay, I think the book told you was the first immunoassay developed, and it was developed to detect the complex of insulin, anti-insulin in diabetic patients. But now we know that it's very useful in detecting small levels of hormones, trace elements really, hormones, some serum proteins, vitamins, things that are really small in size. And the reason that this is such a terrific test method in a lot of ways is that the radioactive label that it uses, I-125, iodine 125, is really easily incorporated into protein molecules by attaching. It attaches to tyrosine within the protein complex, so it's a simple method of labeling. And as such, it makes the test method fairly reliable. It simply emits gamma radiation, which again, simple to read with a gamma counter. You measure the gamma counter and you're going to read it off a standard curve. The level of gamma radiation is relative to the amount of radioactivity that's there. So, it's great. It's very sensitive. It's very precise because we can tag exactly what we want to tag as a reactant in the reaction. All these great things, but it has, kind of that one pesky drawback that, it's radioactive. So radioactivity, this has a relatively short half-life, just under 60 days. I want to say it's 59 and change. So it's as far as radioactive substances goes, it has a relatively short shelf life. 2 months for something that you have to order in and bring in, and it's expensive and it's got to be monitored and all sorts of stuff. It's not necessarily something you want to be dealing with a lot. So the short shelf life is somewhat problematic. But also working with radioactive substances, having, employees work with it on any, kind of routine level. You have to protect the patient, you have to protect the employees. Believe it or not, they are, when they do this, some radioactive substances, not necessarily I-125, but, patients may be involved with this. So, working with radioactive substances, not terrific. Disposing of even low-level radioactive waste is a nightmare. So all in all, kind of a lot of disadvantages that ultimately end up, if we can, outweighing the ease of use of this test. This is a heterogeneous assay. We're going to have some rinsing, and it's a competitive assay. Did I say that at the beginning? I don't know if I did. Yeah, well, it is. And we know it's a competitive assay because it's indirectly proportional to the amount of patient antigen. So what happens? I think I have here we go. So how does this work? Let's just walk through the procedure in its most basic terms. So RIA is as a technique, we're really used primarily to look at antibody levels by using a known quantity of antigen that's been made radioactive. So we're going to we're going to label the antigen with I-125. And as I said, that I-125 attaches into the tyrosine of the antigen. The labeled antigen that gets mixed in with antibody for the antigen. They bind to each other. They make a antibody-antigen complex, and then we wash. And here's where the competition comes in. So then we have adding some patient serum as well. So we have one labeled antigen, we have a reagent antibody, and then we have a patient serum. This is a little different than what we're used to seeing. We usually see these mixed together, right? We'd add the patient serum and the labeled serum. We usually add the labeled and the unlabeled together. But in radioimmunoassays, they actually the patient serum, the question of antibody second. And that's antigen. I apologize. We're doing the same as the labeled antigen. So we're adding patient serum to see if there's antigen present. Should be just patient sample, I think. So in any event, if the patient's what happens is we've just bound off antigen to antibody. This I-125 labeled antigen bound to this antibody is not a really terrifically stable reaction. And in fact, in the presence of patient antigen, the patient antigen is more likely to displace this bound I-125 antigen. It kind of kicks it off the antibody and displaces it with patient. So if the patient, if there was no antigen present in the patient sample, the radioactivity of the antigen-antibody binding would remain in place. So if you have a low level of patient antigen, means that radioactive antigen remain bound to the reagent antibody. So you end up with a high level of what? Radioactivity, right? A lot of gamma radiation. So low patient concentration of antigen means a lot of radioactivity, inversely proportional. More patient antigen, if more patient antigen displaces this radioactive stuff, so we have a lot of antigen attached to this antibody and very little labeled. So you end up with high concentrations of patient sample and low concentrations of radioactivity, inversely proportional. Does that make sense? This is a little bit different test just because we're using the unstable nature of radioactivity in the test. So it's a little bit different than what we've seen before, but basically the same concepts. Inversely proportional because it's a competitive assay. Next, we're going to look at the enzyme immunoassays, or the EIAs. We know because we've done this, so I'm going to kind of rip through these things. EIAs can be competitive or non-competitive. They are using basically the antigen-antibody binding with an enzymatic signal in there someplace to quantify the molecules of interest. We typically, in this case, we're talking about using enzymes as labels that react with that substrate that later gets added to the test. And it's the breakdown, the enzymatic breakdown of those products that ends up being the signal that we see. So, enzymes act on a substrate and they produce a signal. So we're going to talk about, kind of chromogenic, fluorogenic, or luminescent signals. So they can produce color, fluorescence, or chemical luminescence. Typical enzymes include alkaline phosphatase and horseradish peroxidase. There are two others that were in the book that I don't know why I didn't put them here. G6PD, glucose 6-phosphate dehydrogenase, or beta-D galactoside are two other really good enzymes that can be used in these EIAs. Why do we use these particular EIAs? They're said to have the highest turnover or conversion of substrate rates. What does that mean? It means that they're really successful in that substrate enzyme reaction and makes it really easily detectable by whatever method it is, the color change or the signal change. They also have a very high sensitivity, and so they make a really good reactant for the test method. I think at this point we all understand why it's so important to do a separation step. But as a reminder, it's important to wash or separate free analytes because, as we said before, free analytes can still react with with the substrates within the test system, causing a false positive reaction. So it's really important to separate any unbound analytes or reactants. So question 2, how do we perform a separation step in EIAs, and why do we do it? Well, we perform a separation step by doing a wash. That's all you really need to say is we're going to do a wash. And why do we do it? To make sure that we remove any unbound reactants that might interfere with the test method, right? We know that. I think we're good at that now. So we just talked about let's see, we just talked about, we talked about radio, we talked about general information about the EIAs, and now let's talk about competitive heterogeneous EIAs. These we've done. We've just talked about these when we talked about all those competitive assays. So again, we're going to kind of cruise right through. The competitive assays, we understand that they use enzymes with the addition of a substrate, again, to measure and detect the analyte. A couple of reminders for us. Competitive assays use enzyme-labeled antigen to compete with unlabeled patient antigen for those binding sites, those limited numbers of binding sites of antibody molecules that have been immobilized on the solid surface, right? Why do we do, what's kind of our reason for using competitive assays? We use them for small antigens, haptens, and we use ones that are relatively pure because we really want to make sure that we don't have things that are cross-reacting. We want things that are really very clear what it is we're looking for. The test methodology allows for that, allows for a very high degree of specificity. So it's important that, it's an important test for us to be able to do some of those very specific things like hormones and drug screens. Those things are often, they're small or very low concentrations, sometimes very hard to detect. So by other laboratory methods. So as I said earlier in the lecture, because we're not looking for the needle in the haystack, we're going to look for a color change or an enzymatic change. That's much easier to detect than finding a needle in the haystack. So if a needle in a haystack made the whole haystack turn blue, we'd know that the needle was in there, right? Same thing in a competitive immunoassay. We don't have to actually find this very, very low concentration of a hormone to know that it's there. We just have to look for the end product of the reaction. And we know with all competitive assays, they're what? Inversely proportional. We know that for sure. What does that mean? The more patient antigen that gets bound, the less enzyme-labeled antigen can be there, so we have less signal change. So one's high, one's low. If we talk about non-competitive enzyme immunoassays, these are generally indirect. Do you remember what I said about indirect? These are the ones where the labeled reactant is not involved in that initial binding. So these are non-competitive, tend to be indirect. Like competitive, very good sensitivity, really good specificity, simple, very low cost, kind of easy to perform. And we're going to look specifically in a second at enzyme-linked immunosorbent assays, or ELISA testing. Honestly, ELISA testing and EIA are pretty much synonymous at this point. I think that people use the terms when they're talking about ELISA, they might talk about EIA, and vice versa. It's really the most common EIA, and so I think its terminology is becoming kind of blurred. The line is blurred, but they do mean different things. ELISA is very specific in that it is used to measure antibody production. We are not looking again for the antigen itself, HIV, hepatitis B or C, something like that that's hard to detect, and maybe in very low quantities. but it could be hard to detect. It could be hard to not just detect it, but to identify it in a sample. So we don't look for it. We look for the antibodies to that, right? We're going to look for the indirect product of the immune response against those things. Let's take they say it's easily applied to point of care testing and home testing, which is a little scary for some of this stuff, but we'll move on from that. So let's take a look at some of the basics of ELISA testing. And really, this should look pretty familiar to you at this point. ELISA, or enzyme-linked immunosorbent assays, are, again, a type of EIA. It's generally an indirect EIA method. ELISA is generally indirect, where you have you're going to be looking for antibodies in the patient's serum. You can, though, have ELISA tests where antibody gets immobilized and not antigen. But in this figure, let's go from here. And this is analogous again to figure 11-2 in your book, and I think it's, if I'm looking, yeah, it's analogous to the antibody capture of figure 11-2. Antigen gets bound to the solid phase, and remember, we're going to have that antigen is now going to capture the antibody. That's why the book terms it an antibody capture. This illustration, which I liked, this simple illustration, is an indirect ELISA. Why? Because that, again, this enzyme tagged antibody is not involved in the original antibody antigen reaction. So we have bound immobilized antigen for whatever it is we're looking for. Let's say it's HIV. We look for an antibody to it. And remember, red is for the patient. So we had red, sort of, reddish. We had patient sample. It binds to the antigen, patient antibody. What do we do when we get a complex? We wash. Then we go in and we add the reagent antibody, the anti-human immunoglobulin that is labeled with an enzyme. We now have a complex of antigen, antibody, anti-human antibody with a tag. What do? We wash. Then what do? We add a signal. We add a substrate. We look for a signal, and if no patient antibody was bound, so if this step had never taken place, no antibody had bound, then this would have washed away, because this antibody is not to this antigen. This antibody is to this human antiglobulin. This would have all washed away, and all we'd have left in here is the solid surface antigen. So if no patient antibody was bound, never binds here, this never binds, we end up with no color change, right? Let's see. In this case, we could do color change, we could do fluorescence, luminescence, but again, this is, while this is an indirect ELISA, referring just to this the relationship of this enzyme, this tagged antibody to the antigen. This is a heterogeneous, non-competitive assay, so the amount of signal is directly proportional to the amount of antibody, the amount of this antigen-antibody reaction, directly proportional. More patient antibody means more of this tagged antibody binds to it, which means you end up with more of a signal. More means directly proportional. So, true or false? Antigen is immobilized in heterogeneous indirect EIA testing. Well, we just saw this in very great detail. It's true. Although remember, we can also bind up antibody, right? All right, but we're talking about indirect EIA or ELISA testing, so it should be very clear. In indirect, it's antigen. Let's look at capture testing, capture assays, which, God help us, we're using capture again in the book. We had the antibody capture, from figure 11-2, which just told us how the antigens that were immobilized on the solid surface could capture an antibody. We also talked about the term of what we call it when we immobilize an antibody to the solid surface. That's called what? The capture antibody, which are just the same terms in reverse. And now we have capture assays. And these are a sandwich immunoassay. We've seen this illustration before. The sandwich assay really is suited to antigens that have multiple epitopes and multiple determinants. Things that multiple antibodies could bind to, because we're not really looking for this reaction. We're looking to see that something bound to this antigen, right? We're looking to see this antihuman globulin. This antihuman globulin binds to an immunoglobulin which bound to an antigen that have multiple determinants. So it's best suited to antigens like antibodies, cytokines, proteins, tumor markers, microorganisms, especially viruses. And we can use it as a measurement of immunoglobulins, and we can kind of monitor, when we see, for example, when we discuss, kind of what someone's immunoglobulin levels, we can do it through testing like this. Let's see. It's important that the antigen not have any epitopes in common with something else. Microbes often do. Bacteria frequently, they're very tricky devils, and they sometimes mask their identity with having epitopes that are in common with something else. So it's really important that the epitope that we use in this process does not have something in common with something else. Let's see. So this is an indirect test method, and so we can look at the procedure. Let's look at it in just a little different form. This is a little different. It's a little simpler, but conceptually the same idea. So we have the surface gets prepared with the capture antibody that gets bound. We know that. It's going to be in excess of what's needed for the test. And so we have we're going to incubate it with, so we've got the capture antibody here. We had a patient sample. If the antigen is present, it binds to the capture antigen, the capture antibody, or the immobilized antibody. We've got a complex, so we're going to wash. I didn't put the wash in here, but I know you all know it at this point. So after we wash, what happens? We add the antihuman antiglobulin, and because this is direct, we do that. We're going to measure, that's going to attach to another. So remember when I just said back here, it was important that we're looking, that this test method, is used for antigens that have multiple determinants. So this antigen, this patient antigen, by having multiple determinants, we're going to be able to bind to the immobilized antibody as well as the capturing, as well as the labeled antibody. So the second labeled antibody can recognize a different epitope, and then it makes a sandwich. Same thing if we look back here, you can have an antibody get added to this, and then you can bind off an enzyme-bound antibody to it. This is just a more complex sandwich. Second labeled antibody recognizes by a different epitope binding than this one, and you end up with a little sandwich of, bread, jelly, bread. It's a sandwich, so the antigen is bound between two antibodies. We can use this enzyme can be colorimetric and can be chemiluminescent, and again, directly proportional. Is this competitive? It is not. So if it's not a competitive assay, it's going to be directly proportional to the amount of antigen in specimen. Yes? We're going to look at the elusive homozygous EIA in a really brief way. The homozygous EIA doesn't require any washing or separation steps. This is a homozygous enzyme immunoassay. It is not competitive, so it's directly proportional to the concentration of patient antigen in the test solution. And this is interesting in that it's slightly different than what we've seen in the past. What we've seen is, in all of our reactions to this point, is that, antigen meets, labeled antibody and something happens, right? A color change, some kind of signal change. That's the most basic reaction that could take place. We've seen variants of it, but the most basic is, boy meets girl, color changes, we're done. So antibody meets labeled antigen, or no, antigen meets labeled antibody. I guess it could be either way. And color change happens. In this case, the, how can I say this? When the antibody, when the patient's antibody binds to the labeled antigen, it actually binds at a site that blocks the action of the enzyme. So interestingly enough, by forming the antibody-antigen complex, we've now bound off and blocked the enzyme, the action of the enzyme. So that when we add the substrate in to the test reaction, we're not going to get that colorimetric change. We've blocked the enzyme label from being able to act on the substrate. So in this case, let's see. I was looking. Nope, I don't have it. So basically, in this case, a high concentration of patient antibody will block a high number of these enzyme labeled antigen sites, resulting in a very low signal. So it is, though it's not competitive, the result is a high concentration of patient antibody means a low signal. So it's, in a way, it's inversely proportional, despite the fact that it's not competitive. And it's only because we're blocking the action of the enzyme. It's definitely by a different, much like radioactive is, it's a different mechanism, so it's interpreted differently. So you just need to remember kind of what the action of a homozygous EIA is by that blocking mechanism of the enzyme, and I think you can walk it through. Let's see, homogeneous EIAs are less sensitive, but they're pretty easy to perform. And they're used really, again, for some of those low molecular weight things that we think about in serum and urine. Here, hormones, therapeutic drugs of abuse testing, things like that. So we're going to talk for a minute about rapid immunoassays, and we're really going to look at an example of immunochromatography, which I think most people are familiar with, kind of a cassette-based one. These are membrane-based, single-use disposable assays. If you've ever seen a pregnancy test, you'll know that's an example of a immunoassay. We're going to either have antigen or antibody being bound to a membrane. In our example, we're going to see some antibody, and we're going to look for a color change reaction. There could, in some cases, be separate steps that are needed in this. Addition of patient sample, washing reagent, labeled antigen. We might have some wash steps. All sorts of things can happen. But let's look at an example, and I think we'll walk through it, and it'll make a lot of sense. So immunochromatography starts with, kind of this membrane here. And let's identify the sites. So this is the site of the patient sample inoculation to the test cassette. We have what's called the conjugate pad, pardon me, that's embedded with monoclonal antibodies with colloidal gold. and that's embedded in this conjugate pad. We have, continue, we have kind of a membrane here, and then we have 2 rows of antibody. This antibody is a monoclonal antibody to whatever it is we're looking for in this patient sample. So if this was a pregnancy test kit, this would be anti-HCG, right? Because we're looking for the HCG here, where this would be anti-HCG. It would be a monoclonal antibody to that. This is a monoclonal antibody to this monoclonal antibody. So this is an anti-antibody. So this is an anti-IgG that's going to look for these antibodies. And this is really a control value for the test. So let's see how it works, and I think it'll make sense. So we're going to first add the patient sample added to the sample pad on the left over here. Because this is a membrane, it's kind of like filter paper. It wicks along. It starts as soon as it touches here, and it's going to wick along here. And so it wicks along here till we get to this first step, to the conjugate pad, where the patient sample meets the monoclonal antibody with the colloidal gold label. So that's the label. This is a colloidal gold label. They meet up, and if this antibody is a monoclonal antibody against this antigen, HCG, for example, they bind off, and you get, down here, they're continuing to wick. You have the patient antigen attached to the colloidal gold labeled antibody. They continue to wick along this test method by capillary flow until it reaches the first test detection system. This, remember, was the monoclonal antibodies to the analyte in the patient sample. So you can see there's the membrane-bound antibody, and it's binding to an antigen determinant site, and the reagent monoclonal antibody with the colloidal gold label is still attached. So this presence of this entire this entire complex, the presence of this gold will react here and cause a color change in this test line. So that's step 1 to us determining if this is a positive test. The sample, though, more of this has to continue to wick along here and meet the second set of monoclonal antibodies. And this is a control. And so, as I said before, this is an anti-IgG monoclonal antibody. And so it's going to react with these. It doesn't really care if there's any antigen attached to it. It's just really looking for an epitope on this antibody. It's an anti-IgG. And so as it wicks, it has to get to this point and get a positive line as well. Otherwise, it doesn't know that there was something faulty with this antibody at the beginning, or if it just never wicked fully through the test pad. So this could be a falsely decreased result. It could be a falsely negative result. We don't know what's going on. So you have to get a positive result in both of the detectors in order for us to call this test positive. If nothing happened here, you would still have to have a positive control value here, or the test is invalid. This is our control that shows that this managed to wick off in a high enough concentration all the way to the end that it would detect any antigen that was present. Simple, right? All right. Immunofluorescent assays, we're almost done. We're going to look at some immunofluorescent testing later in the semester, but for now, just let's take a quick look at it. Fluorescent immunoassays are really just a different kind of immunoassay. Honestly, the key variable, instead of enzyme, an enzyme that we'll be talking about, the detection method is fluorescence. So we're going to look for, instead of an enzyme reaction, we're going to look for fluorescence. The tests that we use are going to detect the fluorescent compound that is resultant from when a reagent is tagged with something fluorescent, and that binds to, for example, an antigen that was fixed to a cell, to a slide, and then we look at it under a fluorescent microscope. That labeled reactant. Do I have a picture of it? I do. Oh, good. Let me show you this. So we have there's our labeled antibody, and what happens is we have cells that are bound to a slide. We add the patient sample, patient antibody, and then we have this anti-human globulin with a label, a fluorescent label, attaches to the patient. We, put it on in a fluorescent microscope. We kind of bombard it with fluorescent light. This compound, this fluorescent compound, right here, absorbs light or energy at a specific wavelength and then converts it to another light or energy that gets emitted at a different wavelength. And we can collect that emitted light and measure it against a standard curve. Very straightforward. It's nothing fancy about it. Let's see what else we know about this. Oh, that was indirect. So we have a couple of different ways to do it. We just go back to fluorescent in general. So immunofluorescence, so we saw basically how it works, how something labeled binds, gets hit with fluorescent light, absorbs the light or energy at a specific wavelength, converts it internally to energy, which then gets emitted at a different wavelength as light. So as a result, we're looking at fluorescence, and it's a very, very subjective assessment when you're looking under a microscope at how much fluorescence is there. So we restrict this to a qualitative observation. It's present, it's absent. We may even be able to say some things about it semi-qualitatively, but it's really just kind of present or absent. We're going to look at the amount of fluorescent against kind of a dark fluorescent field, a dark field. We use it for identification of microorganisms, things in cell culture, tumor-specific antigens, neoplastic, kind of that new genetically changed material, transplant antigens, and we have two methods, either direct, which is great for antigen detection in bodily fluids, or indirect, which is the more common. And we're going to use that to detect antibody, not the antigen. So this, we're going to look, direct testing looks for antigen, indirect looks for antibody. Let's look at direct. In this case, again, we fix the patient antigen, patient slides right to a slide, add fluorescently labeled antibody to the antigen in question, wash, and then hit it with fluorescent light under a microscope. We're gonna see, that excitation and emission step underneath the fluorescent microscope. And qualitatively define, was it present or absent? Do we see fluorescence or not? We use it for a couple of things, Legionella pneumophilia and chlamydia. That's still not typed right. Chlamydia, trachomatis are a couple of the things that we can look at there. Really rapid identification of microorganisms. Pretty easy to do, but it does require specialized equipment. So it's not every lab does it. Indirect immunofluorescence. Again, in this case, we're going to look for antibody. Remember, indirect was looking for antibody, not antigen. So we had the reagent cells that are bound to a slide. We look and see if the patient has the antibody to whatever we bound here, whatever that antigen is, and then we add, we wash, and then we add labeled anti-human antiglobulin, fluorescently labeled anti-human antiglobulin. Wash, and then we're going to look for fluorescence under a microscope. We use this for treponemal antigens. We use it for antinuclear antibodies, and we're going to spend some time on that later on in the semester. That's a really important diagnostic tool for us. And in this case, the fluorescence is directly proportional to the patient antibody, you know what? It's 1-to-1 and there's no competition, so we know it's directly proportional. I'm going to spend 2 seconds on fluorescence polarization immunoassay. It's used for things, that it can be used for things clinically, drug monitoring, concentration of drugs and hormones that are present in a patient. It's also used in kind of food stuff, identification. So, there's limited use within the lab for this, within a routine clinical lab. Fluorescent polarization immunoassay has the unfortunate acronym of FPIA or FPIA. I don't know. It's a competitive assay, so what do we already know about it? We already know that it's going to have inversely proportional relationship when we're looking at the standard curve. Because there's a competition between labeled antigen, fluorescently labeled antigen, with unlabeled antigen from the patient. Based on that, this is a really interesting way that this works. So that the detection of this is, we're going to again look for fluorescent light that gets emitted from a labeled molecule. But what happens is these antigens, when they're just free and floating in solution, they tumble. And as they tumble, when they're hit by a polarized light source, they emit light, and that at a certain rate. And that when we have those labeled molecules get bound to antibody, they're bigger, they're clumsier, so they tumble less, but they actually increase the amount of polarized light that they emit. So the tumbling is slower, it's less, and they are allowed to emit more light. So, nope, I don't have anything else on here. So let me just tell you. So if we have then, because this is again this is competitive, so we're looking at this labeled antigen competing with unlabeled patient antigen. So to bind off to an antibody. And so if I have more patient antigen bound off to the reagent antibody, and I'm washing away this labeled molecule, there's less of the antigen, there's less of the labeled molecule bound to antibody, so we have less polarized light. We have more polarized light. So whenever you have less of the labeled antigen, because we had more of the patient antigen, we have more of the polarized light emitted. So if we have a low level of patient antigen, means we have a lot of labeled molecules being bound to antibodies. We end up with a lot of polarized light. So low concentration of patient means a high concentration or a high light emittance, a high polarized light emittance. So it's inversely proportional. And then finally, chemiluminescent. This is another antigen-antibody combination. Chemiluminescence is honestly just the emission of light caused by a chemical reaction, and the chemical reaction is typically an oxidation reaction. That oxidation reaction produces an excited molecule that eventually decays back to its original state. That chemical reaction, when it's doing an oxidation reaction, what's happening is the compound is losing electrons. I don't know if I ever told you that my 9th grade chemistry teacher had a saying that honestly I remember 100 years later, and it was, Leo the lion says grr. And this is how I remember all these years later oxidation and reduction. So loss of electrons is oxidation, that's Leo, and then it says GER, G-E-R, gain electrons is reduction. So if you're gaining electrons, it's reduction. If you're losing electrons, it's oxidation. So in this reaction, where there was an oxidation reaction. Did we gain or lose electrons in the chemical reaction that caused this emission of light? We lost electrons, right? Chemiluminescent can be used for both heterogeneous and homogeneous assays, just depending on how it gets set up, and gets used for detection of things like cardiac markers, hormones, and vitamin D levels. And that brings us to our last slide. Yep. So questions 4 and 5 for our class participation. So based on what my science teacher just told me, a loss of electrons is what? Oxidation or reduction? LEO. Loss of electrons is oxidation. Gain of electrons is reduction, because Leo the lion says grr. And that's it. Please come see me if you have any questions, and I will see you soon.